Mitosis is the basis of growth, cell replacements, and tissue repair in multicelled species. Also, asexual reproduction in many single-celled and multicelled species. Meiosis is the basis of sexual reproduction, persuades formation of gametes or sexual spores. Let's start with interphase, where the cell spends most of its life. In the first growth phase, which is called G1, the red and purple inside the nuclear membrane represents the unwounded chromosomes, and the green circle represents the centrosomes. Human body cells usually have 46 chromosomes. Body cells have diploid or 2N, which means that they have 2 of each chromosomes. During the S phase or synthesis, the chromosomes duplicate each other and then create sister chromatids. And then the blue dot represents the centromeres that connect the sister chromatids together. In the last growth phase, which is called G2, it is now ready for mitosis. During prophase, DNA coils tightly to form visible chromosomes. The nuclear membrane dissolves and the nuclei disappears. In the animal cells, the centrioles are not visible, but they separate with their newly formed asteres, which are visible to move each pole. The chromosomes line up at the equator of the cell during the metaphase. One spindle fiber of the conspicuous spindle system attaches to the centromere of each chromosome. The centromeres divide, and the single-stranded chromosomes, now consisting of one DNA molecule each, move to opposite poles. This happens during the anaphase. In the animal cell, cytokinesis occurs with cleavage furrow, dividing one cell into two cells. In plant cells, cytokinesis occurs when the formation of a cell plate. The parent cell now forms two daughter cells.
See, fun!
Meiosis 1 begins with prophase 1 when homologous chromosomes pair up. You have a tetrad consisting of 4 chromatids that intertwine the 2 chromosomes to stimulate the synapsis. This stimulates the process of crossing over and in the result of recombination of genetic information. The crossed over tetrads line up in the center of the cell. The homologous chromosomes separate to opposite sides of the cell. This is the difference between mitosis and meiosis during the anaphase. Centriole duplication takes place in the telophase 1. Formation of the nuclear envelope and the cytoplasm division often occur to produce 2 cells. Interphase 2, it's the amount of time spent rest, and it's also called interkinesis. In prophase 2, the centrioles that are replicated separate and move to opposite sides of the chromosome groups. The chromosomes are centered in the middle of each daughter cell. The sister chromatids separate, and now they are called daughter chromosomes, and they pull towards the opposite sides of each daughter cell, and it separates in the centromere region. The nuclear envelope forms and the cytoplasm divides.
Yummy!
You can now eat.
These chromosomes are delicious!